Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like, share, and subscribe the channel. In the last session, we studied about general reactions of amino acids and color reactions of amino acids. In today's session, we will study proteins. The word proteins is derived from Greek word that is proteus, which means primary. As the name shows, the proteins are made up of paramount importance for biological systems. Out of the total dry weight, body weight, three-fourths are made up of proteins. Proteins are used for bodybuilding. All other structural and major functional aspects of the body are carried out by protein molecules. In this diagram, we can see that proteins perform many essential functions of the body like enzymes in digestive system, muscle contraction, provide support to the body, hormones, antibodies, etc. Abnormality in protein structure will lead to molecular diseases with profound alterations in metabolic functions. Proteins contain carbon, hydrogen, oxygen, and nitrogen as the major components major components, while sulfur and phosphorus are minor components. Nitrogen is characteristic of proteins. On an average, the total nitrogen content of the body proteins is 16% by weight. All other proteins are polymers of amino acids. In this diagram, we can see that misfolded proteins can produce metabolic alterations. Acids are linked by peptide bonds. Alpha carboxyl group of one amino acid reacts with alpha carbon amino group of another amino acid to form a peptide bond or CONH bridge. Proteins are made by polymerization of amino acids through peptide bonds. In this diagram, we can see that carboxyl group of one amino acid reacts with alpha amino group of another amino acid to form a peptide bond or CONH bridge. The two amino acids are combined to form a dipeptide. Three amino acids form a tripeptide. Four will make a tetrapeptide. A few amino acids together will make an oligopeptide, and combination of 10 to 50 amino acids is called a polypeptide. By convention, big polypeptide chains containing more than 50 amino acids are called proteins. In a tripeptide, there are 3 amino acids, but these 3 can be of the total 20 amino acids. Thus, 20 raised to 3 is equal to 8,000 different permutations and combinations are possible in a tripeptide. An ordinary protein having about 100 amino acids will have 20 raised to 100 different possibilities. This number is more than the total number of atoms present in the whole universe. Thus, even though there are only 20 amino acids, by changing the sequence of combination of these amino acids, nature produces enormous amount of markedly different proteins. Structure of proteins. Proteins have different levels of structural organization: primary, secondary, tertiary, and quaternary. Primary structure, the sequence of amino acids in proteins. Primary structure denotes the number and sequence of amino acids in the protein. In this diagram, we can see that there are different levels of structure of proteins. Here we can see that the primary structure of proteins. The higher levels of organization are decided by the primary structure. Each polypeptide chain has a unique amino acid sequence decided by the genes. The primary structure is made by the covalent peptide bonds. The student should have a clear concept of the term sequence. See the following example. Glycine, alanine and valine is the first example, while glycine, valine and alanine is the second example. Both the tripeptides show above contain the same amino acids, but the sequence is altered. When the sequence is changed, the peptide is also different. Characteristics of peptide bond. The peptide bond is a partial double bond. The CN bond is trans in nature and there is no freedom of rotation because of the partial double bond character. The distance is 1.32 angstrom units, which is marked midway between single bond, which contains 1.49 angstrom units, and double bond, which is 1.27 angstrom units. In this diagram, we can see that proteins form partial double bond where the distance is 1.32 angstrom units, midway between 1.49 angstrom units and 1.27 angstrom units. The side chains are free to rotate on either side of the peptide bond. The angles of rotation, known as Ramachandran angles, therefore determine the spatial orientation of the peptide chain. Dr. G. N. Ramachandran did pioneering work on the structural aspects of proteins and during 1950s and 60s. In this diagram, we can see Ramachandran angles which determine the spatial orientation of the peptide chain. Numbering of amino acids in protein. In a polypeptide chain, at one end there will be free alpha amino group. This end is called the amino terminal, that is N terminal end, and the amino acid contributing the alpha amino group is named as the first amino acids. Usually the N-terminal amino acid is written on the left hand side when the sequence of the protein is denoted. Incidentally, the biosynthesis of the protein also starts from the amino terminal end. The other end of the polypeptide chain is the carboxy terminal, that is C terminal, where there is a free alpha carboxyl group which contributed to the by the last amino acid. In this diagram, we can see that N terminus is first amino acid and C terminus is last, that is fourth amino acid in a tetrapeptide. All other alpha amino and alpha carboxyl groups are involved in peptide bond formation. Amino acid reduces in polypeptides are named by changing the suffix -ine to -il. For example, glycine to glycyl. Thus, peptide bonds formed by carboxyl group of glycine with amino acid or group of alanine, and then carboxyl group of aniline with amino group of valine, and is called glycyl aniline and valine, and abbreviated as NH2 DGLY ALA VAL COOH, or it can be abbreviated as GLY ALA VL, or simply as GAV. Branched and circular proteins. Generally, the polypeptide chains are linear. However, branching points in the chain may be produced by interchain disulfide bridge. The covalent disulfide bonds between different polypeptide chains in the same protein, that is interchain, or proteins portions of the same polypeptide chain, that is intrachain, are also part of the primary structure. In this diagram, we can see that cysteine forms disulfide bonds between two chains of insulin. Rarely, the instead of alpha carboxyl group, the gamma carboxyl group of glutamic acid may enter the peptide bond formation. Example, glutathione, that is gamma glutamyl cysteine glycine. The term pseudopeptide or isopeptide is used to denote such a peptide bond formed by carboxyl group other than that present in the alpha position. Very rarely, protein may be in a circular form. Example, gramycidine. In the diagram, we can see that glutathione utilizes gamma carbon instead of alpha carbon in gamma glutamyl cysteine glycine. Primary structure of insulin. As an example of the primary structure of protein, that of insulin was originally described by Sanger in 1955, who received the Nobel Prize in 1958. Insulin has two polypeptide chains. The A chain, that is glycine chain, has 21 amino acids, and B chain, that is phenylalanine chain, has 30 amino acids. They are held together by two interchain disulfide bonds. A chain has 7 cysteine and B chain also has 7 cysteine, which are connected. Similarly, A chain has 20th cysteine and B chain has 19th cysteine, which are also connected. There is another intrachain disulfide bond between 6th and 11th cysteine residues of alpha chain, A chain. The species variation is restricted to amino acids in the position 8, 9, 10 in A chain and in the C terminal of B chain. The amino acid sequence has been conserved to a great extent during evolution. In this diagram, we can see that insulin has two chain disulfide bonds, one chain intrachain disulfide bond, and A chain starts with glycine, whereas B chain starts with phenylalanine. The porcine insulin and human insulin are structurally similar, except the amino acid in B chain, that is threonine, is replaced by alanine. Bovine insulin may be produced produced antibodies in humans by repeated injection. But dealienated porcine insulin bear no antigenic sequence from insulin because insulin will not produce antibodies in the diabetic patients even after long-term use. Nowadays, human insulin is being produced by recombinant DNA technology. Here ends our session. Kindly like, share and subscribe the channel. Thank you.